students, in the last video we have learned that how the dorsal ventral axis as well as right and left axis is forming in C. elegans. Today we are going to see the gastrulation process in C. elegans. What is gastrulation? Gastrulation is the process of forming three germ layers like ectoderm, mesoderm, and finally endoderm. Ectoderm that will give rise to the skin and nervous system. Skin and nervous system. Mesoderm give rise to the muscular system, cardiovascular system, and the reproductive system. Endoderm will give rise to the lining of the lungs and the gastrointestinal tract. This is just for your information. Okay. So, the process of formation of those three germ layers, that is gastrulation. In the case of C. elegans, when this gastrulation starts, after the formation of P4 cell, that is after the fourth division, and after that also that embryo goes on dividing, and as it reaches its 24 celled stage, that stage onwards gastrulation starts. Here we can see that 24 cell stage, and in that stage there will present a fluid filled cavity inside this embryo. Its name is blastocele. Okay. Uh, and we need to familiarize all the cells present inside the embryo. As we learned in the last class, we know that these cells are the AB cell descendants, these are MS cell descendants, these are E cell descendants, these are C cell descendants, and this is D, and this is the P4 stem cell. And what would be the first process that happen in the gastrulation of C. elegans? First process is the migration of daughter E cells from the ventral side towards the inner side of the embryo, and it divide there and will form a gut primordium consists of 20 cells. As a result of that process, that blastocele, it reduced to a pit. Its name is blastopore. Blastocele get reduced to a pit. Its name is blastopore. And to this pore, those cells that is destined to move inner side of the embryo enters. Clear? I will show you what is blastopore. Blastopore is a pit that found in the side of the embryo, and through that pit the cells fated to be endodermal. They can leave the outer surface and enter the embryo. So that is blastopore. Then what is the second process? Second process and is the movement, is the migration of P4 stem cell through the blastopore towards the inner side of the embryo. It moves inward and occupies a position below the gut primordium. Okay. And the third process is the migration of MS descendant cells. MS descendant cells through the anterior side of the blastopore towards the inner side of the embryo. Followed by that, the C blastomere descendants as well as the D blastomere descendants. They also migrate to inner side of the embryo through the posterior side of the blastopore and enters internally. And after that, that we can see here, it's not correctly, I can't give that animation. The C cells also enter the embryo internally. And after 6 hours of fertilization, after this migration of MS cell and the CD blastomere descendants, the AB derived cells. We know that these are AB derived cells. And among them, those AB derived cells destined to form the pharynx, they move to the inner side of the embryo. Okay, clear? And some AB descendant cells will left there. They undergoes a special type of movement. Its name is epiboly. Epiboly means all these cells, they develop some pressure, become flattened. It become flattened and spread and cover all the other layers in this way. It covers and ventrally at a point it will, sorry, ventrally at a point this that AB derived cell can meet. I'm just drawing only the last point, last portion of that cell movement. And here the two end will meet, and that meeting point is sealed by E-cadherin. E-cadherin. Okay, by that way, this AB derived cells form a layer around all the other layers outwardly and will form, this will form the ectoderm. Okay, clear? This will form the ectoderm. And next, during the next 6 hours, that cells again continues the same pattern of movement and develop into organs. And later, that ball-shaped embryo stretches out to become a worm with the 558 somatic cells. And additionally, 115 cells also is forming there, but they undergo apoptosis. They are destroyed. And after that, the worm undergoes 4 molts and becomes sexually mature, a hermaphrodite adult. And it contains 959 somatic cells as well as numerous sperms and eggs. By that way, it completes its gastrulation. Okay. Now, with I will show you some pictures of the gastrulation in C. elegans. In this picture, we can see that the blastomia descendants have almost reached the inner side of the embryo, and because of that, blastocyst have reduced its size. In this picture, we can see the 3 germ layers: ectoderm, then mesoderm, and finally the endoderm. And once more, I am telling that the ectoderm is formed from AB cell descendants, and the mesoderm is formed from that MS cell and C and D cell descendants. And finally, the endoderm is formed from the E cell descendants. Okay, with this, I hope that everybody have understood the gastrulation in C. elegans. Thank you.
I'm an icon, man. My girl about a 10. I should fuck her in a Benz.